Okay, I hope you can hear me now.
Yep, we can hear you fine. And we're still looking at your PowerPoint. Yes, okay, good.
Okay, thank you.
Perfect.
So I would like to start by thanking Tom for the kind invitation to speak in this interesting symposium. It's a pleasure, and what I'm presenting here is basically work we've been doing in the regulation of social behavior using zebrafish, but that is relevant for stress. So the first part of the talk is basically to establish the link of the signaling pathway that we are interested in, which is oxytocin, and then we'll move from that to the role of oxytocin in modulating the way social environment can modulate the stress response. So starting with neuropeptides and their role in social behavior. Repeatedly across evolution, these nonopeptides have been implicated in the regulation of different social behaviors, even in invertebrates. This is a trend, and with the gene duplication and the evolution of these other paralog ligand, oxytocin became more free, so to say, to respond to selective pressures, and became consistently associated with one kind or another of social behavior across vertebrates. In terms of studying social behavior, one of the issues that usually we come across is the fact that social behavior can mean different things. And so I think it's important to try to explicit what social phenotypes we are addressing, and the two most elementary social phenotypes that one can consider is the willingness to be with others, that we can call social motivation, and basically it's can be measured by approach withdrawal tendencies of the individuals. And then a set of social cognitive abilities that organisms need to navigate their social lives. And social attention and social memory are the most basic ones, so that they can that they can tell individuals apart and behave differently towards different individuals in the group, and so basically individualize relationships across individuals. And in zebrafish, we have recently shown that from a phenotypic perspective, and looking into how these different behaviors cluster together, that you can see different behavioral modules organized, with the motivational and the cognitive module being separate. This is relevant if we want then to consider the mechanisms involved in the evolution of these different modules. In this same work, we also found, by doing a custom-made SNP array for a list of candidate social genes that we collected from the literature, we also found a number of different genetic variants, different SNPs associated with these different dimensions of sociality. And among them, it's oxytocin. So although oxytocin, as I was praising before, can be seen as a genetic toolkit, it's by far not alone in controlling the expression of these behaviors. There's a whole suite of other variants and genes contributing to variation in these behaviors. So as I was saying in the beginning, I will first talk about how oxytocin modulates these dimensions of sociality, and then we'll move on to the most relevant part for this audience of how it regulates the use of social information to modulate response to stressful situations. So in terms of the contribution of oxytocin to social behavior, what we can say so far is that, to start with, social behavior is something that at least zebrafish is not born with. So it's something that develops along the ontogeny. And they become social more or less around 3 weeks of age. And we say that based on their preference to stay near others that you can see illustrated here in this left panel. And because it develops during ontogeny, one question we had was if the role that we have been ascertaining to oxytocin could be a developmental role if oxytocin is not an activational signal in the adult brain, but if oxytocin is needed to basically establish the neural circuits that are needed to implement adult social behavior. So we did this together with the lab of Gil Lefkowitz, and this was a work led by a former postdoc in the lab and Rita Nunes. What Rita and the team did was basically to use conditional ablation of oxytocin neurons using a GAL4 UAS line, which has an oxytocin promoter, and then it has natural reductase attached to the UAS, such that the activation of the nitroreductase would drive the silencing of the cells that have that promoter. Basically, would silence oxytocin neurons. And so this is a way of silencing, of ablating oxytocin neurons in a conditional way, basically at the time that one wants to do it. And so we did it at different points in development between 4 and 6 days old, individuals 12 to 14, and these two data points are before, if you remember, the 21 days, they become social, and then we did it also once they already became social. And so the idea here was to look for a critical period in development in which oxytocin would be needed to implement these neural circuits needed for sociality. And the results are summarized here. And what you can see here is that oxytocin has this significant effect in adult behavior if the neurons are ablated before the individuals became social, but not after they became social. And in adulthood, it had no effect. So this is highly suggestive of a developmental effect in a critical period when they are not yet social, but they are on their way to becoming social. So then the most obvious thing to consider as a mechanism underlying this effect was that we were depleting the brain of oxytocin neurons. So we counted the number of oxytocin neurons, and a bit to our surprise, they come back. And so the ablated individuals don't have this behavioral deficit because of a shortage of oxytocin neurons. Oxytocin neurons apparently are doing something else during that critical period, most probably regulating some other circuit in the brain that becomes affected. So we looked into the dopaminergic system because it is well known that oxytocin talks to dopaminergic clusters in the brain, and the two together act in concert to regulate different behavioral phenotypes. And so we looked both in larvae and in adult fish for the effect of this ablation of oxytocin neurons in the numbers of dopaminergic neurons in different clusters. And there were 3 clusters in adulthood that showed significant reduction in the number of dopaminergic cells, and they are basically involved in different aspects of dopaminergic function. So you have the cluster 7 in the pretectum that is involved in the processing of visual information, and you have these ventral clusters 11 and 12 that are more involved in the role of dopamine as a rewarding signal. And so basically what apparently is going on is that the silencing of oxytocin at early age is basically modulating the circuitry of these dopaminergic clusters, such that in adulthood you have shortage of dopaminergic cells, both in the pathway that processes visual signals, and one can speculate social visual signals, and also in the pathway that is involved in reward processing. We then established that there was a physical link between the oxytocin neurons in the POA and these clusters that we were seeing depleted in cells. And so that was done with another construct with synaptophysin that allowed to establish the occurrence of these cell-cell contacts between projections of oxytocin neurons and these dopaminergic cells. And then we moved on to see, okay, once there's these structural effects, are also functional effects of these that parallel the deficit in behavior. And so we counted the number of cell nuclei in terms of activation using the expression of PS6, a ribosomal protein that becomes phosphorylated when neurons are active. And basically what we found was significant differences in two different areas, in the anterior part of the preoptic area and in the anterior part of the VV, which is, I would say, an homologue of the lateral septum globally in a mammalian brain. And so these two areas showed parallel changes in their response to social stimuli in the individuals, in adults that have been ablated as young individuals. We also looked into the overall pattern of functional connectivity across the brain, and we did this by basically doing network analysis of the PS6 data. So one can look into co-expression patterns, and from there, with network analysis tools, basically build a core network with simulation statistics, and then analyze that network in a number of network metrics. And what I'm sharing here with you is basically the effects of exposure to a social stimuli, both in the control and treated animals and in the ablated animals in the bottom two figures. And so what you see is basically an integration of an increased integration of the network in the controls with more links to other nodes outside the local communities being established. And you see the opposite basically in the treated animals, in the ablated animals, where you see basically a segregation of these different modules in the brain. And this is a visual representation, but obviously the network statistics support this conclusion. And so in summary, in this first part, what we are showing is that oxytocin is acting on social behavior by modulating dopaminergic circuits during development. And these dopaminergic circuits, once they are kind of crystallized in development, they are relevant for the implementation of adult social behavior in such a way that later manipulations on the oxytocin no longer have an effect. Despite the fact that we are showing these very specific effects of oxytocin in development of sociality, we are very much aware that these complex phenotypes are typically not genetically determined. So what we also did was to try to see how the role of oxytocin can be modulated by the environment in which the animals are being raised. And so we raised, in this case, oxytocin receptor mutants in different social environments and looked for these two different types of phenotypes in adulthood. And what we found was that there were gene by environment effects on social behavior. And without getting into details, because I think that the most important part of the talk is the one that follows, what I would like to leave here as a take-home message is that despite these very large irreversible effects of oxytocin on the development of social behavior, you still see to a certain extent rescue of these phenotypes depending on the environment in which the animals are raised. And typically in husbandries, you raise mutants together for convenience, and part of the effect of on the behavior that you see on the mutants does not come from the mutation in the target gene, but from indirect effects of this mutation on peers with whom they are socializing. And this is just a summary of what I just mentioned to you that I won't repeat again. And I would now move on to the second part, more related and to the stress. So the idea to do this was, okay, so we have oxytocin that is involved in the regulation of social behavior, and individuals can use others as sources of adaptive information, namely as let's call them sentinels, to detect danger in the environment and basically help to reduce vigilance and allow individuals to do other things in life and rely on each other for the detection of danger in the environment. And we decided then to see if this type of phenomena was present in zebrafish. We took advantage of the fact that in zebrafish, as in other fish, skin extract that basically signals a wounded individual in the environment elicits a stress or alarm response, whatever you want to call it, that has this active phase with the zigzag swimming, followed by this immobility phase with freezing. And this is a very stereotypic behavior that can be easily quantified with 1 or 2 parameters, in this case, position in the water column and speed over time. And you can see here where the both parameters change. That's when alarm substance was introduced, and you see that they decrease speed, and they also decrease position in the water tank and come near the bottom. And this is elicited by the alarm substance that I mentioned to you, that is released from these epidermal cells. And taking advantage of this, what one can do is now to study how the social environment is modulating the response to these adversive or aversive stimuli. So we studied different aspects, social buffering, in which our target individual receives information directly about presence of danger in the environment by giving them this alarm substance. And we can have them smelling the alarm substance, but observing a shoal of conspecifics that are not alarmed. And so we would expect some kind of buffering occurring in this situation. The opposite, that is contagion, where our focal individual is not smelling directly the alarm substance, but we give the alarm substance to the neighboring shoal. And so although they don't smell danger themselves, they see their neighbors alarmed and expressing these alarm responses. And then social facilitation, that I will not address today, where you give the alarm substance to both, and you see if the presence of alarm conspecifics enhances the alarm response in a focal fish that is already smelling danger. So we started by searching for this phenomenon in zebrafish, and we found it. We found that they basically reduce, as you can see here in these two bottom graphs, their response to the presence of alarm substance in the water if they smell the alarm substance in the presence of relaxed conspecifics. And this effect lasts in time, at least up to 30 minutes. Then we tried to figure out which sensory modalities were involved, and we just gave them either visual access, chemical access to the conspecific signal, or both. And obviously, the highest reduction you see is from both, but any of the sensory modalities is efficient in buffering the response. In other words, even if you don't see conspecifics, but if you smell relaxed conspecifics, you don't react as your stress and alarm response is not as high as if you are by yourself. And to our surprise, we did not see effects of shoal size in this response. And so 2, 4, or 8 individuals were equally efficient in buffering the response. Then we moved on to test the social contagion. For doing this, we did what I just described to you, and we showed that indeed, if they are observing a shoal of alarmed conspecifics, they will also express alarm, stressful behavior. And then we decided to test the role of oxytocin in these responses. And the reasoning was, well, since oxytocin is modulating these elementary behaviors, such as preference and recognition, these are behaviors needed to perform more complex social behaviors, like the use of social information for adaptive decision-making, and therefore oxytocin can be involved. So here we took advantage of a set of CRISPR mutant lines for the ligand and the 2 receptors. And what we did, and there's a lot of information here, but if we focus in the second column of graphs, it's probably the most informative and easier to follow. It relates to freezing, and what you can see here in the, for example, the oxytocin receptor knockout, the gray bars show an increase in the freezing when they are exposed to conspecifics that are alarmed. But in the orange bars, you don't see the same thing in the mutants. So in other words, the mutants are not responding to the presence with alarm conspecifics by expressing alarm behavior themselves, whereas wild types do. So the presence of social contagion in wild types is basically cancelled in the mutants. The same thing happens to the oxytocin receptor-like knockout. So zebrafish, because of the duplication of genes as these 2 genes for the receptor, and the same thing is seen in the ligand knockout. The interesting thing one can do with ligand knockout is to go beyond the role of oxytocin as necessary for social contagion, because now we can treat the ligand with exogenous oxytocin and try to see if one can rescue the phenotype and see if it's not only necessary but sufficient for social contagion to be expressed. And that's what we did. And basically, as you can see here, you see the reduction and you see the recovery in the treated ligand knockout, indicating that you can recover the phenotype by giving oxytocin back to a oxytocin depleted system. We then looked into the brain to try to see where we could find parallels of these behavioral effects of oxytocin on social contagion. And again, VV popped up. So this is the same area that I mentioned before. And in this case, VC was also another area that showed a similar pattern. And the interesting thing that you can see here in terms of activation is that whereas in the wild types, what you see is a decrease in the activation of these areas. The pattern is the same for both ventral areas of the pallium. So there's a decrease in activity in the wild types when they express this social contagion. And in the mutants that do not express this social contagion, what you see is the opposite, is an increase, a very large increase in activity in this area, which suggests the presence of an inhibitory circuit that we subsequently validated by using also transgenes for the VGLUT and the GAD markers of glutamate and GABA, and confirm the GABAergic identity of most of these cells. We also looked into projections of the oxytocin neurons into these ventral telencephalic areas. So the area where VV and VC are located are more or less here. We have also marked in red the dopaminergic neurons to help us locate these areas because they are known to have projections there. And you can see here in green the cell bodies of the oxytocin cells, and I hope you can see these lines coming forward into the forebrain, and you have an amplification here in the bottom. And these are the projections of oxytocin neurons that bring the oxytocin here into this area where we are seeing the effect. And finally, we just did some quantitative PCR to confirm the expression of the two oxytocin receptor types in these areas. And as you can see, and I just alert you that you have very different scales in the two graphs, so please don't compare them directly one to the other. But both the oxytocin receptor and the oxytocin receptor-like are expressed in the VV and VC, which are the areas where we see the effect. So finally, what was still missing to close this story was, okay, so we see our focal individuals responding to changes in the stress or alarm or fear behavior of others. How are they doing it? Are they simply imitating, in terms of motor imitation, the behavior of others, like we do with yawning? Or is this more than that? And basically, they are responding to changes in the internal state of others that is signaled by their behavior. And so to disentangle these two types of explanations for these, Kyriakos, the postdoc, and I forgot to mention that the work I was describing until now was the PhD work of Ibukun Akinrinade, the former PhD in the lab, now at Yep Payne's lab in Calgary. And the next piece of work was then followed up by Kyriakos in the lab. And Kyriakos developed this setup of disentangling of, separating in time the moment when our focal fish observes the demonstrators and the moment it reports a behavior to discriminate between demonstrators. And so we had two screens. In one screen, we would display a demonstrator that was always relaxed. In the other screen, the demonstrator had 3 episodes of alarm response. And then in a second point in time, in the test probe phase, both demonstrators are relaxed. So if you find a difference in this second phase, it cannot be explained by what our observer is basically seeing at the moment. It needs to be a reflection of what he observed in this previous time when one of the demonstrators was expressing stress. And make a long story short, we don't see a very significant effect on attention. We replicate the contagion that was observed with real demonstrators. This is reassuring that the video playbacks are working, because you can also see the same contagion when video playback stimuli are used. And then the critical part of this study is the test phase. That's when they have to basically respond to demonstrators that are currently doing exactly the same thing, but that were seen before doing different things, meaning one was stressed and the other one was not stressed. And what you can see here basically is that while wild types discriminate between the two demonstrators and, to our surprise, express a preference to go towards the individual that they had seen before in distress, the mutants do not do that and do not express a preference. So apparently, wild types are able to recognize that an individual was seen in distress at a given point in time, and oxytocin mutants are not able to perform this operation. And so, to summarize, we show that contagion and also buffering occurs in zebrafish, that oxytocin is necessary and sufficient for fear contagion, that this is associated with the decrease of activity in an inhibitory population of GABAergic neurons in the basal forebrain, and that the role of oxytocin in the regulation of fear contagion is implicated in, I would say, either emotional recognition, or, if you find the word too strong, recognition of an internal behavior in others, and not simply based on motor imitation. I would close by basically highlighting the people that did the job. So the first work, as I mentioned, was the work of Ana Rita Nunes, former postdoc in the lab. The second part was Ibukun Akin Rinade, former PhD, and the follow-up study was done by Kyriakos Karekos. And this is work done in collaboration with the lab of long-term collaborator and friend Gil Lefkowitz at the Weizmann Institute in Israel. So I finish here. Thank you for your attention. If there's still time, I will take questions.
Thank you very much, Rui. So, are there any questions for Rui? We have a few minutes. Okay, Kei Yamamoto, I have to allow you to ask the question, so I'm the one slowing things down now. There you go. Go ahead.
Should I?
Hold on, you're still muted. And yes, please, and stop sharing. That might make it easier to see each other.
Yeah, that's what I was going to say.
Do you hear me?
Yeah,
yes.
Yeah. So I come back to the question about the dopamine neurons. So you said that there's an interaction with the dopaminergic cells of posterior tuberculum and also pretectum with the oxytocin neurons.
Yes.
But like we discussed after the first talk, that the reward system related to the reward system. This, I think, we kind of consensus that it's an old idea and not anymore. And the first question is, so you looked at all the cell population, dopaminergic cell population, and you see specifically in the posterior tuberculum?
Yeah, so
okay.
we screened for the different dopaminergic clusters, and these are the three clusters where we found an impact of the ablation.
So two posterior tuberculum and the pretectum.
Yeah.
Okay, because it is interesting because the pretectal cells also in mammals, there are no dopaminergic pretectum cells.
Mm-hmm.
So they are rather non-homologous cells,
mm-hmm.
playing the kind of the same role. And with this, I would rather interpret as a convergent evolution instead of conservation as a system. You know, I wouldn't doubt that oxytocin cells are there, but how it works, it may be rather convergent than conservation.
You are raising an interesting question. That is the question of homology in behavior and neural circuits, and at which level you establish, and if different levels of biological organization can, if you can have homology at one level that requires or not homology at a lower level. And these are the discussions of the 90s with George Strider and Glenn Northcutt.
Yeah, exactly. So as a system, because all the claim of this social behavior, social brain network. When I see each nuclei in what is claimed as conserved in teleoth, I don't see conservation. You know, the pretectum, that, like you said, and the posterior tuberculum, dopamine cells, preoptic area, the anterior preoptic area, it's not conserved because in fish, this project directly to the hypophysis, and this kind of projection we don't see in mammals, because in mammals and birds, it's project to the median eminence, and it's a portal system which bring the peptide to them. So
that's the issue I was mentioning. That depends at which level we will address it. Is it cell type? Is it circuitry? Is it neuromodulator? So what we are showing here is that oxytocin itself has a conserved role. The way it is implementing this conserved role, meaning the circuits in which it is acting, are not necessarily conserved. But apparently the same molecule is performing the same role and affecting the behavioral phenotype. But as you were saying, probably through different arrangements of circuitry, motives, and that involve the connectome, the variations in cell types. That's another thing that we are far from understanding. We talk about these neural regions, but the characterization of cell types at each of these regions is not very clear in comparative terms. We know it for very few animals so far, mostly mammals, but even cell type homology, it's an open question.
But at the system, I'm quite convinced rather that it's, the co-opting several times, the same molecule somehow giving the same, similar output, the outcome.
Yeah, sure.
Okay. Well,
all right.
It was interesting.
Thank you very much. Spock, do you have a quick question before we move on? Because we're slightly over time, but I know we have some flexibility on the break.
It was a quick one. I was really fascinated by the recovery of phenotype that you saw in the knockouts, and I'm kind of curious: is there a non-social teleos that we could kind of test this with? Let's say, can we recover something that may have been lost, ecologically? The reason I ask this is because I've seen, for instance, in the Panthera, which are the big cats, we have lions that are the only social ones, and tigers and leopards are asocial, but I assume the system is conserved there. So if you applied oxytocin, thought experiment, can we recover it?
Well, this would be pure speculation. But if you could do a CRISPR lion or something like that, it could be possible. I think this has been tried with voles and mice, and you're probably aware of these kind of monogamous mice and story and so on and so forth. So I would say it should be possible, and it's a fascinating question. It's more a question of finding the right model, either in terms of a natural system or a model organism where one can do that. But certainly that would be fascinating to do.
Great. All right. Thank you very much. Thank you, Rui.
Thank you.
And then our final speaker before the food break, as I call it, is Karina Santiago Gonzalez, who is a PhD student in my lab and who's going to present